A monoclonal antibody is an antibody made by cloning a unique white blood cell. All subsequent antibodies derived this way trace back to a unique parent cell. Monoclonal antibodies can have monovalent affinity, binding only to the same epitope. In contrast, polyclonal antibodies bind to multiple epitopes and are usually made by several different antibody secreting plasma cell lineages. Bispecific monoclonal antibodies can also be engineered by increasing the therapeutic targets of one single monoclonal antibody to 2 epitopes. It is possible to produce monoclonal antibodies that specifically bind to virtually any suitable substance. They can then serve to detect or purify it. This capability has become an important tool in biochemistry, molecular biology, and medicine. The role monoclonal antibodies have come to assume was first postulated by early 20th century immunologist Paul Ehrlich, who proposed the idea of a medical Zauberkugel, a magic bullet. This would be a compound that could be made to selectively target a disease-causing organism, which would allow a toxin for that organism to be delivered along with it. He and Elie Mechnikov received the 1908 Nobel Prize for Physiology or Medicine for this work. In the 1970s, the B-cell cancer multiple myeloma was known. It was understood that these cancerous B cells all produce a single type of antibody. This was used to study the structure of antibodies, but it was not yet possible to produce identical antibodies specific to a given antigen. 324 production of monoclonal antibodies involving human-mouse hybrid cells was first described by Gerald Schwaber in 1973. This work remains widely cited among those using human-derived hybridomas. In 1975, Georges Kohler and Cesar Milstein succeeded in making fusions of myeloma cell lines with B cells to create hybridomas that could produce antibodies specific to known antigens and that were immortalized. They and Niels Keijer shared the Nobel Prize in Physiology or Medicine in 1984 for the discovery. In 1988, Greg Winter and his team pioneered the techniques to humanize monoclonal antibodies, eliminating the reactions that many monoclonal antibodies caused in some patients. In 2018, James P. Allison and Tetsuko Honjo received the Nobel Prize in Physiology or Medicine for their discovery of cancer therapy by inhibition of negative immune regulation, using monoclonal antibodies that prevent inhibitory linkages. Much of the work behind production of monoclonal antibodies is rooted in the production of hybridomas, which involves identifying antigen-specific plasmablast cells, makes them unable to use the de novo pathway and become fully oxotrophic for nucleic acids, thus requiring supplementation to survive. The selective culture medium is called HAT medium because it contains hypoxanthine, aminopterine and thymidine. This medium is selective for fused cells. Unfused myeloma cells cannot grow because they lack HGPRT and thus cannot replicate their DNA. Unfused spleen cells cannot grow indefinitely because of their limited lifespan. Only fused hybrid cells referred to as hybridomas are able to grow indefinitely in the medium because the spleen cell partner supplies HGPRT and the myeloma partner has traits that make it immortal. This mixture of cells is then diluted and clones are grown from single parent cells on microtiter wells. The antibodies secreted by the different clones are then assayed for their ability to bind to the antigen or immunoblot. The most productive and stable clone is then selected for future use. The hybridomas can be grown indefinitely in a suitable cell culture medium. They can also be injected into mice. There, they produce tumors secreting an antibody-rich fluid called ascites fluid. The medium must be enriched during in vitro selection to further favor hybridoma growth. This can be achieved by the use of a layer of feeder fibrocyte cells or supplement medium such as Bricklone. Culture media conditioned by macrophages can be used. Production in cell culture is usually preferred as the ascites technique is painful to the animal. Where alternate techniques exist, ascites is considered unethical. Several monoclonal antibody technologies have been developed recently, such as phage display, single B cell culture, single-cell amplification from various B-cell populations and single plasma cell interrogation. Technologies. Different from traditional hybridoma technology. The newer technologies use molecular biology techniques to amplify the heavy and light chains of the antibody genes by PCR and produce in either bacterial or mammalian systems with recombinant technology. One of the advantages of the new technologies is applicable to multiple animals, such as rabbit, llama, chicken and other common experimental animals in the laboratory. After obtaining either a media sample of cultured hybridomas or a sample of ascites fluid, the desired antibodies must be extracted. Cell culture.